is computational biology. And the idea is that let's compare, let's say, biology to physics or maybe even maths. And both in physics and maths you have a very formal language to express your ideas, right? But sometimes it happens so in biology that since biology is very fuzzy, uh, your ideas are mostly qualitative rather than quantitative. It becomes sort of like, okay, this protein is present in some, let's say, twice the amount of some other protein in normal conditions. So very likely this is going to have this effect or something. The problem being that since you want to have a quantitative understanding of things rather than a qualitative understanding, what that will allow you to do is make a prediction, right? So the idea that science can make predictions based on our uh, based on our already existing knowledge and
Hey, your volume is going really down. Uh,
okay, is it
yeah, better now.
uh,
Yeah.
wait, let me change my input volume a bit. So let me tell me if it gets too loud. uh, is it much better now?
Yes, yes, very much.
Okay, okay. So yeah, so I was saying that computational biology helps you to qualitatively rather than qualitatively guessing and making a hand wave argument, give a quantitative understanding. The problem becomes that there is too much quantitative data in the current uh, sequencing. If you have followed any of the current COVID research, you would know that all sorts of genomes have been sequenced. Every variant has their genome sequenced. People have sort of figured out which protein interacts with what, and all the protein structures are mostly known. So there is a huge explosion of information, and this has led to new fields which have emerged. And as science progresses, biology is also adopting techniques from other fields and sort of growing as it progresses. So I will first tell you about a field which is known as systems biology. I will show you a very simple example of a research paper, what they did, and sort of give you the background of what is the logic behind what they are trying to do. And then I'll talk slightly about the kind of work I'm doing. I can, for that one, I can show you some of the data which I currently have. And then maybe we'll talk about some ethics and maybe also some other experimental biology techniques, which are also quite fascinating. Okay, so without further ado, let's share my screen and let me know if it's visible. Take a, uh, is this showing? Hello?
Yeah, we can see it now.
Yeah, yeah. Is it, it's legible, right?
Yeah, it's legible.
Okay. So what we see is this is, as you can see, this is a very simple Boolean system. And let's say that what we have is three variables, let's say A, B, and C. And we have states of these variables. They can either be 0 off or 1 on. And based on uh, some logic, the value of the state, these variables A, B, and C will be decided in the next step. So let's say A is going to be and of B and C. So here both B and C are 0. So A stays 0 in the next step, something of this sort. And what you can do is you can, so this is a work of Stuart Kaufman. You can look him up. And this sort of idea that you can use Boolean networks like this to model biological metabolic networks was his pioneering work. So Kaufman was a medical student by training, but the time at which he was earning his PhD was a time when computation, like the use of computers was exploding. People were gradually getting access to more and more computational power, and that sort of encouraged him to take this sort of an approach. And one of the primary reasons he took this Boolean approach is that this is quite simple to understand, in the sense that you don't have, like, you have only just two states, either it's off, either it's on. And also, given the computational power, this was the sort of the most easiest model to run and get an understanding of stuff. So what you can see here is that if you start in, let's say, a state 110, so A in the next step is going to be 1, and you can just check these rules. So these states are going to fluctuate one after the other. But there are some other states like 000, which is self-perpetuating. So if you are in this state, you keep staying in this state. But there are some other states like the 010, which also sort of gather a lot of other states. So these states, which are sort of the attractors for multiple states, are called basins. And it's not necessary that a basin has to be just one state. It can be like this. This is a two-member basin. This is a one-member basin. This is another one-member basin. Is this part clear? Does anyone have questions still here? Okay, so let's go ahead and let's see what sort of biology we can get out from this. So don't bother about the names of these proteins. Take a, what you are seeing here is this is a very simplified network of proteins which are important for cell division in yeast. Okay. And what we can do is experimentally people have shown that this protein can inhibit the function of this protein. If it's a red arrow, it means that this protein inhibits the whatever arrow it's going to. If it's a green arrow coming in, it means that this protein activates this protein and increases its function. So from experimental data, this was known that this is going to activate this, but to what extent it is going to activate, that was not known. So in presence of that sort of data, it's easier to make a Boolean model where you just say that, okay, just the presence of this is going to activate this. And what you then try to do is you use, this is a, whatever variables there are, for them you use a Boolean model and you try to see which states are acting as basins, which states are sort of very transient states. And if you do that, so I'll just show you what the table shows here. So there is a state which looks like this, in which some protein is on and some other protein is on, every other protein is off. And as you can see, 1764 nodes lead to this state. So there are some 11 variables. And since it's a Boolean thing, you have some 2 to the power 11 states, which is 2048. And then here, this adds up to 2048. You can check. So of 2048 states, a majority of states given will fall into this basin. Okay. And if you see that this state corresponds to a state called G1 state. So a refresher for those of you who have like sort of forgotten some parts of biology. So this is the representation of the cell cycle. So a cell's existence is sort of to replicate. Okay. So the cell, what it's going to do is it's going to take nutrition from the environment, and it's going to grow in size. And that phase is called G1 phase. After it has grown to some size and it knows that it has got sufficient nutrients, only then it starts replicating its own DNA. It starts replicating its uh, protein uh, protein amounts. So it sort of now has double the amount that it began with. And after all this synthesis has been done, the cell again takes some time to check that, okay, all the synthesis has been correct or not. It sort of runs an internal check. And after the check is done, it then proceeds to mitosis, basically cell division. So it's a very concerted process and a very cyclic process as well. And as you can see, most of the states, it doesn't matter what initial states they begin at, they're going to come back to this G1 state. So that what it tells you is that the cell cycle is extremely robust, even when you use a very simplified version of the model. So in this model, what they have done is, uh, so there are multiple checkpoints here during this process where the cell can stop if it wants to. What they have done here is they have removed these checkpoints. So this is a very simple system in which if once the cell size is reached, it doesn't care, it's going to complete the full cycle anyway. So in a very simplified system in which you take a very simplified model, you can sort of replicate what nature has evolved and perfected over millennia. So just to show, this is sort of the network of their study. Okay. And you can see that there are some, uh, okay, in this figure, there are no such states. This only shows the all the 1748 states which are leading down to this. This is the G1 node basically. Okay. So this type of understanding also has a very interesting aspect. So in this thing, in this figure, what you can see is we are looking at the system as a whole. We are not just looking at a very specific protein and what changing that protein is going to do. And so biology, as I mean, I have some exposure to biology, so I can tell for biology. Biology has been a reductionist science for most of its uh, development. Okay, so people, let's say, would be like, okay, I just want to understand only this very specific gene, and that has led to some very important developments. We have understood which genes have what functions. But if you look at a disease, let's say, let's look at cancer, let's look at diabetes, it's not just caused by one gene, it's caused by a combined effect of multiple genes. And in that situation, just having a very reductionist approach sort of breaks down because you lose sight of the larger picture. Maybe we can take also the example of ecology. So ecology, if you try to understand the whole ecosystem, you can't just look at a single lion or a single deer in a large forest, right? You have to try to understand what a whole population of this lions are going to do, what a whole population of this deers are going to do, and how their interaction is going to model the ecological landscape of that particular forest or national park, whatever. So systems biology allows you to, so this uh, discipline is called systems biology, and it allows you to have a very bird's eye view of biological systems. So here we have looked at maybe a cell cycle network. You can apply this similar sort of principles to maybe metabolic networks. And a large portion of this advance in this field has been driven by a person called Uri Alon. So he was again one of the pioneers in this field. And as you can already see that this correlates very well with uh, graph theory and stuff, right? So a large number of people who sort of come to this sort of metabolic modeling field or systems biology are from maths background. People from even physics background come in because they sort of uh, maybe understand mathematics better than biologists in some sense. Okay. So this part is for systems biology. I have tried to show an example. If any of you have any questions, let me know.
I think some of them did not quite understand it in detail. Do you
Okay.
want to perhaps like summarize what you just said for a complete layman to biology in terms of what systems biology implies?
So what systems biology tries to do is sort of look at, let's say, whole metabolic network, or maybe a whole group of proteins at once, and the goal being that if we look at this entire group of proteins or maybe genes, whatever, at once, we will understand the basically biology better. You can't take a cell, let's say, and just say that just changing this one protein here is going to affect the cell in this way. Okay, if you make a very drastic change that this protein is required for some very specific function, yeah, it's going to maybe kill the cell, but it doesn't tell you much about how this protein had interacted with other parts of the cell. To understand that, you have to sort of zoom out. Rather than looking at this single protein, you have to look at groups of proteins which are now interacting amongst each other. And only then do you get a holistic understanding of the entire process which is happening. So this is what systems biology tries to do. Is this clear?
Yeah, I think that was a clear summary. So you're basically saying that, computational models are able to, in some sense, replicate these processes or interactions
Yes.
between smaller components
So
in biology.
here, so in this study, what they did was they took a cell cycle kind of a thing, right? So these proteins control the cell cycle of so cell cycle is basically this of yeast. And just, uh, they were able to show using a simple Boolean model like this, that any state you start in, you are very likely to end up in a G1 state. So basically, let's say you are currently in mitosis, that cell is going to go again and start dividing. You start in some other phase, that cell is again going to come back to this dividing, like pre-division state. So the cell is already primed, always primed for division, and that this network is robust. If the network is not robust, let's say a significant fraction of these nodes would have like just gone to some other node, and they would have maybe made a separate basin or a separate cycle. So the cell would be stuck in, let's say, some state, and it can't leave, so it can't divide anymore. Okay, something of that sort. But that doesn't happen in nature because this entire system is quite robust. And thinking about it, this is yeast. So yeast has somewhere around 800 genes. Humans have somewhere around 20,000. And it's kind of been shown that the large number of genes is from larger number of regulatory elements. So humans have more proteins which sort of take care that nothing goes haywire. Okay. Is this part clear, or should I?
Interesting. Yeah, that's interesting. So that happened over evolution, right? And
Over evolution, yeah. So it's sort of like nature has got enough time to run optimization algorithms, kind of in that sense. Because if
oh,
you are dead, you just go out of the equation. So it's a very strong optimization in a sense.
okay. Is there a direct correlation between the number of genes and the complexity of the
No, no,
organism?
no, no.
Okay.
It doesn't work that way. Actually, I can tell you, onions have more genes than humans.
Oh, okay.
Uh, but that's partly because most plants have something called polyploidy. So they have larger number of, uh, they have multiple copies of the same gene. And sometimes, since we are talking about complexity, systems biology also looks at complexity. And biologically, it's better to, rather than just, let's say, looking at three genes or whatever the number of genes, it makes more sense to sort of understand it from a network's perspective. Let's say you have five genes taken, and this is how they interact with each other taken. And let's say I have four genes, but they interact like this. They have like cross connections. So one may argue that this is a more complex system rather than this simple five gene, like just pentagonist thing. Okay? Because this has potential for more dynamic responses. Is this part clear?
Yeah.
Any questions? I please feel free to ask.
Someone says also more number of chromosome pairs does not mean more advancement in the
Yeah,
in
it of course it doesn't.
okay.
And again, like uh, there is a correlation with genome size and uh, like complexity. If you call like uh, let's say humans more complex than, let's say, yeast, something of that sort. But it's that there are always going to be exceptions in biology. You can't run away from that, because some animal somewhere taken on evolutionary time scale has found some other optimized niche in which no other animal has gone. So they have like some strange genomic whatever. So you will always find that in biology. But overall, you can say that there is a correlation, but uh, the correlation shows up more in network topology rather than just the genome.
Got
So,
it.
okay.
Cool. Yeah.
So this part was some part of, let's say, systems biology. And so the idea here is looking at systems as a whole. One may have asked here that at what point do we go from being reductionist to, let's say, more looking at stuff holistically? Because even if we are looking at a metabolic network, we can't model the entire cell yet. We can model only small parts of the network. So it sort of becomes, again, one can argue that it's also reductionist, but it's a process. You can't just go to the very reductionist level and straight up jump to the entire holistic level. You sort of have to gradually build up, and that's what currently the endeavor is in this field. Okay, so that being said, let me talk about some part,
Uh,
somewhat.
just one question before you
Yeah,
move on. So,
sure.
since we were already talking about complexity, um, so how, in physics, you have systems that are predictable and deterministic, right?
Sure,
So,
sure.
when you're talking about systems biology, it seems like there are a lot of factors and parameters in play. So, would you
Yes.
say that biological systems are deterministic? And if not, like what level of uncertainty are we usually talking about in even in small systems like the example that you gave?
So if you look at biological systems, they are fuzzy. Fuzzy in the sense that you will have some bound of fluctuations. Okay? But it won't happen that you suddenly leave this bound and completely go to some other place. If this is happening, you are either shifting to some other state or the cell is likely going to die. Okay?
Okay.
So the, as you can see, as you increase the number of like interacting bodies. So let's say, as a simple example, a two-body system is easy to solve. You insert a third body, and it becomes quite difficult to solve, right? There are only uh, there are some, you have to make some assumptions, and only then you get some stable orbits for a three-body system. So biology is also sort of like that. You have to have some parameters in parameter range in which your cell has some functionality, cell or protein, whatever. But if you sort of go beyond this range, you run into problems. The cell will sort of lose its integrity. It will maybe die. It may become cancerous, something of that sort. So there are studies on Boolean networks and cancer. Um, I can't remember the person's name, but so what he works on currently is this metabolic networks in cancer, and he tries to show that if you do a Boolean modeling like this, uh, where did it go? Yeah. So this study, which has a very prominent basin, so cancerous cells instead have multiple basins, and since the cell goes and sits in those basins, it can't escape from a like non-physiological, like a non-healthy, let's say, a non-healthy state, and it gradually becomes cancerous. Taken. Was that clear?
Yeah, yeah, thank you.
Okay. So going ahead, so this also I'll give some brief introduction to the protein which I'm working with. So let's say we have some genes. Let's say we have a gene A and we have a gene B, and for some reason we want this to be present and we want this to be absent. So in cells, the way this is achieved is something called uh, you sort of silence this gene. And the one way to do that is put a protein here, okay? And in my case, this protein is named CTCF. This is just a name. Don't attach any value to this. So, so this protein, what it basically does is that it will now silence this. The way it can do this is it doesn't allow the transcription machinery, basically the machinery which converts from DNA to, not converts, which creates an mRNA from a DNA template. So it basically doesn't allow that machinery to bind here. Okay. And it is known that presence of CTCF can convert genes or portions of DNA into something called heterochromatin, which basically means that it's very densely packed DNA. And since it's densely packed, it has low levels of transcription. You don't create as much protein or mRNA. You don't create as much mRNA. Okay. So that makes CTCF a very important genomic regulator, and some mutations in the portions in which CTCF binds sort of reduces the effectiveness of CTCF binding. And if this binding efficiency is reduced, in some cases, you can have situations in which this gene B is expressed when it shouldn't be expressed because now CTCF can't bind. This protein is not present. And this has been known to be factors in multiple sorts of diseases. CTCF actually stands for 60 CTCC binding factor. Okay, there is a this whatever name, and there is a region of DNA CTCCC which this binds to. In any case, so the thing which I do is molecular dynamics. So the way it is done is uh, there are many proteins for which structures have been solved, and there is a database of these solved structures. So let's look at four. I think it was HHB or something. Yeah. So I'm showing you this structure because this was one of the this is one of the pioneering studies which was able to solve the structure of a protein. Okay. So X-ray crystallography in its sort of infancy, and they were able to solve a full protein structure here. So this is basically, as you can see, it's deoxyhemoglobin, and you can see that there are some four colors. So this purple, some red, some orange, and some green. So they're all four subunits of this protein. Okay. So basically, what this database has is the location of each atom in this protein. And what I can do is that I can download this structure and just a second. This is sort of the structure I'm working with. Let me zoom in a bit. So this double helical thing is of course DNA, as you know. And what you can see here is, okay, this is too janky. Just a second. Let me fix my mouse sensitivity. Okay. And the thing in white is actually a representation of the protein. So this representation is called a cartoon representation, and it's easier to sort of visualize. So you can see that there are these coils, okay, and then there are these sort of two arrows. So those are some specific protein structures, and what I can do is I can take this entire structure, initial structure, which is present somewhere in PDB, and then I can sort of simulate how this protein is going to behave over time. So I have rather than looking at a very static picture here, which just shows you a snapshot, and the way X-ray crystallography is done is that this has been crystallized, it's present in some concentration of salt. All those also have an effect on protein structure. So just to understand how the protein is going to behave in normal bodily pH and water, I'm not showing you the water. There is water surrounding this protein. So you basically take this entire structure, you simulate it over time, and you try to look at the dynamic nature of this DNA protein interaction. So just a second. So this is the simulation over time. I will just, so as you can see, it sort of has some fluctuation. So it's a dynamic structure, and you sort of you don't have this dynamic nature of, you can't see this dynamic nature of this protein DNA interaction in the PDB, just the basic structure. And once this sort of structure has been done, you can sort of gather some data from it, and it becomes yeah, some quantitative data from it. And I will show you some representation which is done. So as you can see here, there are these repeats, right? So let's say this structure, and then this bands, and then this repeats 3 times. Is this visible? Can you see this?
Yeah, it's visible.
Yeah. So as you can all see, there is a repeat of these 3 units. And if you go back to the structure, you will see that there are actually 3 units of this as well. So there is this unit here, another here, and another here. So this representation is like human readable, but this is more machine readable. And you can use sort of representations like this to again write machine learning algorithms as well. So I think you have all heard of AlphaFold, uh, which uh, which was making a huge amount of news a few months back. Uh, do you, any of you? No one? That's surprising.
Yes, yes.
You have heard? Yeah. So basically, what the DeepMind was able to do in Google, basically, that they were able to write a machine learning algorithm which sort of uh, can tell you the structure of a protein just from its sequence. You And that's a very uh, large jump in the accuracy of the structures generated as well. So that sort of allows you to like sort of entirely skip X-ray crystallography or whatever. Crystallography is a very tedious business in biology. You have to wait for the crystals to form. You have to find the exact conditions in which crystals are going to form. You can sort of now bypass that part if you have like faith in machine learning. I'm not saying that like crystallographers are entirely out of business, but they are facing some very stiff competition right now. Okay. So this is sort of the stuff that I currently do for my research. I'm trying to sort of look at, uh, let's say, I have a DNA sequence which is non-mutated, and I can sort of plot this sort of a contact map. This thing is called contact map, and I can now create mutations in this DNA and look at how that specific mutation is changing the contact map over time. So maybe there is some mutation which reduces the amount of contacts. Maybe there is some mutation which uh, enhances the contacts, and from there one can sort of get an idea that given some genomic sequence, which proteins are going to be expressed, which proteins are going to be uh, like repressed. Okay. Okay. Any questions from this part?
Do you personally work with these machine learning models as well?
I haven't started working on them, so I'm
Mhm.
currently generating this data.
Okay.
Then, so currently, what I can get sufficient information about this binding and stuff from just looking at the energy of binding. But but the thing is that here I know what mutations I have created. If I want to completely automate the process, right, then what I will try to do is maybe do mutations in some other locations and get some training set, like mutations here in this location of the DNA has this sort of an effect. And maybe one can try to sort of write an ML algorithm to sort of guess what's going to be the effect. And depending on the amount of training it does, again, it's going to have better and better predictions. That's like quite, even if it arrives, it's going to arrive quite late into what the project is currently leading to. Currently, it's sufficient amount of information can be done just from basic analysis.
Interesting. So, the mutations that you were talking about, I think even yesterday you mentioned,
Yes.
you specifically study mutations caused by a certain source, right? Not by, there are multiple sources of mutations, if I'm not wrong.
So what you can do is you have this. So, as I said in the very beginning, next generation sequencing has led to a huge amount of data being generated. So what you can do is you can look at the genomes of animals, and you sort of have an idea. Uh, this is again a very interesting field, bioinformatics. So bioinformaticians can like they have algorithms which can sort of say that okay, this protein is going to go and sit and bind to this thing, and that way you can identify CTCF binding sites, and then you can look at the sort of variants of this, like whatever consensus sequence is available, and then you can look at what mutating each residue in each location is going to do. Some locations are, if you look at, let's say, uh, let's take a random DNA sequence. Okay? Let's say it's A G T C C G A, and I have the same sequence in maybe let's say four animals, and it's like C G E A G A, and something else, let's say P G A A G A, something of this sort, and you can already see that there is some residue, let's say this G, which is same in all of this samples, and GA here is present in all samples. So from this, I can sort of construct a consensus sequence kind of a thing. And I can say that whatever is the length of this uh, sequence, there is going to be a T here, and there is bound to be a GA here. If it has to bind to some specific piece. Sorry, this is a G. Okay, all this information bioinformaticians can provide. And then you can sort of go and look at, okay, why this gene? What makes it so special? Then you can go and take a look at the structure, and then you can think maybe let's change this to C. What happens? So this is the sort of idea behind doing this also.
Okay. Okay. Got it. There was just one question that I'd like to go back to in the chat box.
Yes.
Nature has had almost infinite time to run its optimizations or find the right gene out of a huge search space. So
Mm-hmm.
how do biologists or rather computer scientists even attempt to replicate it on their puny computers? Um,
Uh-huh.
Maybe I'll just add to that. So basically, I think, is it possible, first of all, to model natural selection? And when you're talking about mutations that happen perhaps naturally in the course of time of evolution,
Sure.
is it possible to model such things, such
So
optimizations? Yeah.
a brute force, you can just brute force it, but that's obviously impossible, right? Because a human genome is some what 8 megabases. Megabase is basically maintained to the 6 base pairs, and if you want to do a mutation for each of them, so the kind of simulation which I showed you just now, it takes somewhere around 29 hours to run. Okay, and you can the larger your like whatever number of atoms you're simulating, this is mostly an order of n square algorithm which you have to use. The reason behind it being that what you have to do is, uh, okay, bear with me, I'm not very good at physics. So if you have the initial structure of the protein and whatever protein DNA stuff, you have some sort of a potential energy which you know, and you can set a fixed temperature, okay, for your system. So this gives you some sort of a kinetic energy. And what now you do is basically, given this initial conditions, what is going to be the state of the system at the next time step. So you increase the system by a very small fraction, very small time step. Let's call it tau. Okay. And this is at t. So at state t plus tau, you sort of, depending on this, you apply basic Newton's laws of motion. You sort of find that, okay, it's in this some state. And once you are sitting in this state, now you have to again calculate the potential energy of the system. To do that, you have to calculate the pairwise distance for all atoms, which is basically n square. So this limits your algorithm to order of n square. Okay. And as you can already see, if this is an order of n square thing, and you're doing this for all genomes, this is not going to happen.
Right.
So simply speaking, we can't do what biology has done, what nature has done over the years, okay, over some billions of years. But what we can do is, rather than blindly trying to brute force it, as I said, bioinformaticians who are mostly from computer science backgrounds, actually, the algorithms which have been designed, people from computer science backgrounds have been, like, or even mathematics backgrounds, have been extremely, you know, have been pioneers in this, designing algorithms for this, and they can sort of give you some regions of interest, like this region in all across multiple animals maybe has seen very low change. So that gives you an idea that if this had been changed, it sort of likely kills the animal. So you only see only the animals which are surviving currently, they all have the same variant. You can have some other proteins or some other genes, whatever, which show very high degree of variation, which you can guess maybe it's happening because selection pressure is acting heavily on this gene, and nature is sort of trying to find the best possible, candidate. So there are all sorts of variants, and they all have some survivability potential, but gradually over time, one of them is going to sort of become dominant and win over the others. So both of them have been found. There are genes like this. Uh, so some genes like ribosomal genes, they are very stable throughout evolution. There are some genes like cytochromes, which have seen so much change that there are cytochromes which across animals which don't even look the same. They are completely different proteins. The only similarity being that some amino acids in their active site, like where the enzymatic reaction is going to happen, they align similarly. So what nature is trying to preserve there is just this geometry. Anything else doesn't matter. So all sorts of stuff you can find. And uh, truth be told, our computers can't be classified as puny anymore. We have quite good computational power, and of course, not everybody can avail it, but we have the potential to come close to, like, mapping many things on, like on the gene.
Right.
But of course, I mean, trying to beat nature is a, high expectation currently, at least.
Yeah. On that note, like, I mean, we're talking about like at the DNA or the protein level, but
Mhm.
do you want to comment on like the evolutionary algorithms that are,
Oh
quite
yeah, yeah.
popular and
Good.
in
So,
some sense do mimic evolution?
so you're talking about genetic algorithms, stuff like that, evolutionary
Yeah,
algorithms,
yeah, that allow you to, kind of control the
kind of
mutation
control the
rate and things
mutation.
like that. Yeah.
I actually have. Wait, uh, I don't know whether I can run it on this one. Just a second. Uh,
But if anybody else has any questions
yeah,
so
yeah. Please.
far, or,
It's been,
you know,
I would like to have a more discussion.
yeah, any comments also, some interesting thoughts you might have, please, feel free to unmute and talk.
So this is actually some Python code which I had written. So what this tries to do is, there is a given polynomial, okay? given polynomial which looks like this: 3 minus 4.3x plus 5.9x squared plus 5.2x cubed plus x to the power 4 and equal to 0. What I've provided the algorithm, okay, is that it knows the any what I've provided is that given any initial, given this equation, okay, I have plugged in values and I have given it the values at like at -5, the equation has this value. At -4, it has this, something of this sort I have given. And then I have told it that you can choose coefficients in some range. Okay. I think the range for this is between -20 to +20. Whatever. So I've given it some range, and then what it tries to do is, let me go back to this writing pad. What it tries to do is, let's say it generates a whole group of, let's say this is the original plot, okay, original polynomial. What the algorithm is going to do is it's going to first generate all sorts of random polynomials, okay? And what it will measure is the least square error, and it will sort of grade this generated polynomials based on how low this least square error is. And it will select the best of these ones and it will try to, combine it basically. So it's sort of like a reproduction thing with the other best ones. And it will gradually try to fix the, uh, you know, fix the basically coefficients.
Hmm.
Let me see if this runs properly. So this, so you can see gradually it's trying to improve, right? This lines which are building up, you can see them
Yeah, yeah.
gradually this band. So the original one was the one in red,
Okay.
and this blue one is the best one for the first when everything was randomly generated. And over
Wow.
generations is trying to sort of find its best match. Okay. Uh, as a
Interesting.
Yeah, it's still, I think, running. It takes some time to sort of find its best match, and once it does, it gives the coefficients on this plot. Whatever. So this is sort of this algorithms are called genetic algorithms. They try to mimic this sort of things. Uh, there are some YouTube videos as well where people have tried to um, sort of mimic altruism, how altruism emerges uh, in case any of you don't know altruism. Altruism basically means what bees do, right? Uh, there is a queen bee, and only the queen bee has the right to pass on its genes. The worker bees don't pass on their genes directly, but they still sort of help maintain the hive, help maintain the health of the queen, and like they fight for the hive and stuff. So what is the benefit, one may ask, that why do this bees don't they just leave and create their own colony or something of that sort? Yeah, so basically it has found the best match. Okay. uh, it took some 15,000 iterations in this case. So the black line is the best match, and this is the least square error. So the when it started out, maybe it had a large least square error, but I have fixed that once the least square error goes below 0.005, it's pretty much good enough for all practical purposes. So this is like this was actually the one of the first Python projects I did. This is a like revised version, but and a revised and improved version. But a genetic algorithm was the first Python project I did. Okay, in any case,
Wow.
so this is a very interesting part as well. So this sort of most evolutionary algorithms fall into the uh, this optimization type algorithms. If you ask that whether we can sort of model evolution itself, that's a tricky question, and people have tried. There are some game theory aspects. People have tried to model evolution via game theory. People have tried to sort of see how altruism can evolve over time. People have tried to look at how maybe even so we are social animals, right? how does this communal thing gradually evolve over time? People have tried to do all sorts of things, but they're mostly math heavy, and I don't understand much math, so I don't understand that good enough to at least give you a perspective. But you can look these things up for sure. Okay. So I think we are currently at 9:30. Uh, so I think
Yeah, we can definitely have a part two of the discussion.
sure.
So maybe today we'll just stick to whatever we've already spoken about in terms of this. And would you want to perhaps summarize or, talk about, just mention the different categories that biology is tending towards today, and especially some of the fields that involves other fields like mathematics and physics?
Okay. So there is something called now, which is quite uh, like gaining momentum, is something called single molecule biology. So the idea here is you can now look at the level of a single protein. Maybe you can sort of understand what it's doing. So I can maybe give you two examples of this. Uh, so let's assume that you have somehow made a track, okay? And on this track, you can walk a protein, uh, and this protein's function is to carry cargo inside the cell. Okay. So what you can do is, let's say you have the protein, which for some reason looks like this in this example, uh, and it carries some cargo. And so I'm basically telling about Fruit Malik's work. Okay. You can look him up. He is currently at IIT Bombay. Okay. And he uh, he is from a physics background. His doctorate was in physics, and then he shifted to biology. And the kind of biology he does is this: you can sort of have a cargo which you feed to the cell somehow. I'm not going into the details, which looks like a sphere. Okay, and this is basically sort of it acts as a lens, and if you shine laser through this, maybe I'll disable here. The scale of this cargo is so small that if the laser even cargo moves a bit, the laser goes off focus, and sort of it wants to come back to its initial this condition. So this protein, if it walks in any direction, which is not its this stable uh, center of this cargo, it's going to feel a push, and you can sort of now measure the kind of force this protein is generating just based on this behaves like a spring. So you can just use F equal to minus kx. The k you will have to figure out from the laser intensity and all, but that's calibration. Let's not bother about it. And you can sort of, this entire thing is happening under a microscope. So counting the pixels, you know x, and you can measure the force which each protein generates in real time. That's amazing. Okay. For biologists, it's amazing. I mean, and there is another experiment which was, I think, this was a 2016 science paper. What they did was they took, uh, let's say, a protein which is unfolded, and they put three fluorophores. So let's call this F1, let's call this F2, and let's call this F3. What is special about these three is they all show basically there is a phenomenon called FRET. The brief idea is that you have a fluorophore which has an absorption spectra and emission spectra. So this is for both F1 and F1. What happens is the absorption spectra for F2, uh, let's call this AB and E. The figure is getting quite complicated, but in any case, the absorption spectra for F2 coincides with the emission spectra for F1. So if this fluorophores are sitting far apart, they will just fluoresce. Uh, let's say you excite F1, you will see the fluorescing wavelength of F1. But let's say now what has happened is F1 and F2 are pretty close together. in some range. What will happen is F1 will absorb, it will emit, but F2 will pick up this entire emission and F2 will now emit. So instead of seeing the fluorescence of this molecule only, you will see some fluorescence from this and a large amount of fluorescence from this. So this thing is called Forster resonance energy transfer or fluorescence resonance energy transfer. Both of them are fine. And what these people are able to do is, since this protein is going to fold, this F1 and F2 and F3 are temporarily going to come closer, move away, come closer, and all that, right? So they're able to, from this, looking at the basically FRET. So let's say they're exciting only F1 and they're only looking at F2 emission spectrum. So when F2 is present, you now know at which time points these two regions in the protein are close together. So you can see a protein folding in real time. All this is possible currently in biology, and all this ideas, actually these ideas are from physics people, right? All the fluorescence, physics, chemistry both, right? And biology is sort of now becoming the most interdisciplinary field you can find. Everyone can apply something in biology. People try to flock towards it. You can find very fascinating, basically, applications of multiple fields in biology. Okay. Anything else?
Thank you. That was a fascinating example. Anybody from the guided project here who wants to talk about ecology, perhaps, or um, any questions from the project that you're doing currently? Also, there's one question, I'll just rephrase it
Yeah.
quickly. If you could, restart the life on Earth, would it take the same evolutionary pathway that it has already taken? Basically, if you run it multiple times, does it tend to evolve in the same way?
That's a very difficult question to answer. And to be told, I have no clue. Uh, us being here is, a very serendipitous. Very few planets have life to begin with, and
Yeah,
and we are mammals, right? If that whatever asteroid didn't drop on the dinosaurs, maybe we'll just be small marsupials at the moment, right? Or, or maybe if like the cyanobacteria hadn't evolved, we will still be all in the ocean because there is no oxygen. You don't have efficient energy generating processes. We just
right.
depend on whatever diffusing oxygen. We are all in the ocean as non-sentient cells, or maybe even sentient cells. Who knows? I have no clue. So this is a very difficult question to answer. and and
Yeah.
uh, and the problem is that we don't have any way to answer. We can't create our own planets currently,
Hmm.
so we can't run that experiment even to sort of have any idea.
Yeah, I think the fact that it is serendipitous probably means that if you ran like the whole process again, it would be
Yeah,
dramatically different.
it's going to be dramatic,
So,
very likely.
yeah,
I would believe that it's going to be different, but you never know. What if like it's deterministic, right? Who knows?
right. Um, okay, so I think there was rapid evolution in the ancient times, but has it stopped now? Or rather, why has the human body
No.
stopped evolve? Okay.
So human body actually hasn't stopped evolving. We can see some like you can see distinct facial features across different regions of humans, right? That's
Hmm.
sort of what evolution is doing. Uh, the certain in certain localities, those features are maybe beneficial, and they sort of carry those features. If you There are studies which have been which have shown that people in high altitude regions, they have a different version of hemoglobin. By version, I mean basically some residue, some protein has some
Hmm.
mutation. Hemoglobin has some mutation, which allows them to retain more oxygen.
Wow.
So
Okay.
they sort of have adapted. They have evolved, kind of. So one problem which some people have is evolution always seems to you know trigger this thought that they have to speciate. You need to have two species at the end of it, but that's the end maybe that's not even the end goal. That's one snapshot of the process where these two species have emerged. But evolution is not just a snapshot, right? Evolution is a continuing process. So humans are also consistently evolving. Another example is lactose intolerance. It's hypothesized that since European populations were heavily dairy dependent compared to southeastern populations, which who sort of did not consume that much dairy products, instead relied on maybe fishery or agriculture. Uh, European people have more lactose tolerance. There is a enzyme called lactase, which babies produce, which allows them to break down lactose. As you become an adult, the levels of this enzyme go down in your body. For Europeans, it goes down much slower, and in some cases, they retain baby levels throughout. So
Oh, that's
that
surprising. I was of the opinion that, Europeans have uh, lower levels of tolerance,
no,
but
some populations do, but that's again like some distribution you will always have, right? Europeans are
okay.
generally more lactose tolerant. On
Oh,
the other hand, Asian people are more alcohol intolerant. So there is an enzyme called alcohol dehydrogenase, which is lower, which has been shown to be somewhat lower in them. So this sort of distribution is there.
okay. There's one
It,
question.
it's just that it happens so slowly, we think that we are not equal. Because again, evolution brings that snapshot mentality in most cases. Like it somehow feels like that. But you have to think of it as the whole process which is happening.
Okay. Sankarshan, I hope that answers your question. Okay, Akhil has asked, how close are we in creating efficient mutant enzymes to degrade plastic polymers that can be scaled up?
Mhm. So there has been some research. I haven't been following it, and the problem is that metabolic pathways, as I showed, uh, modeling them and like you can have an enzyme which sort of degrades plastic. The problem is that if you try to put this into an organism,
Hey, I think we lost him. Are you guys able to hear him?
Yes, yeah. Yep.
Okay. Rani, we are not able to hear you. I think we lost you for a minute there. Are you there? Okay, I think he lost connection perhaps. We'll just wait for a minute.